Dr. DiPersio presented some, I would say, amazing preclinical data that might lead to a new CAR T-cell based therapy for T-cell leukemias and lymphomas. John, can you tell us where you got the idea for this and how you took that to where it is now?
Yeah, so actually, I'm not sure where the idea came from, but we knew that when we looked closely at T-cell, we wanted to stay away from B-cell malignancies because there were so many people working in this field, and so many, yeah, and so many people had made such progress that we thought that our additional efforts would be at best incremental. So we thought we'd focus on these diseases that nobody's really had a good eye on, and one of these areas of research interest has been T-cell malignancies. And as you know, these are bad diseases. Either up front, T-cell lymphomas are much worse than B-cell lymphomas. And T-cell ALL, although it's sort of similar in outcome to de novo B-cell ALL, once you recur, there's really no targeted therapies for either kinds of either lymphomas or T-cell ALL. So we thought we'd focus on that. But when we looked at potential targets in T-cell malignancies, we couldn't find a single target that wasn't equally expressed on an effector T-cell, the T-cell you would use for a CAR, and the malignancy. So we thought, what if we just genetically, gene editing is something we were doing in other projects in the lab, and we thought we could maybe manipulate T-cells. We didn't think we could manipulate them as efficiently as we have, but we thought it would be a start, and maybe we could learn how to do it better over time. It turns out that we really, it was much more effective than we thought, using CRISPR in particular. And once we identified a particular antigen, and there were a small list of candidate antigens, we chose the one that we knew would be best suited for a T-cell. In particular, if you lose that antigen, the T-cell still functions. And so we chose CD7 because it was the most overexpressed antigen in all these malignancies, and also on all NK cell malignancies. And it's also an antigen that we had known from previous work in the field and our own work. When you delete it from a mouse, with the T cells are a normal number and normal subset and normal function. So we thought that if we deleted that target in the effector T cells and then used those cells as a source for CAR Ts against a T cell target, CD7 in particular, then we would have a winner, if you will. And so it turns out that that does work very well. If you get the CD7 out and you can efficiently delete it biallelically in 95% of the T cells using laboratory or scale-up procedures. You can then put a CAR to CD7 in these cells, and these cells respond beautifully when you direct them towards T-cell malignancies that are expressing CD7. So the thought is that that was one hurdle to overcome, and the other hurdle was getting past this allo issue, in that T cells and T-cell malignancies, they look identical in a patient, and so it's really hard to separate the normal T cells from the malignant T cells. And so to get around that, we devised a way to use an off-the-shelf reagent and disarm the alloreactive T cells by eliminating the TCR alpha subunit, so that the TCR is no longer expressed on the surface. And those T cells expand perfectly fine when they're signaled through the CAR, but they don't have any alloreactivity. So you can, in theory, use anyone's T cells for treatment of T-cell leukemia. You just have to delete the target, CD7, and the T-cell receptor, and put a CAR to CD7 in them.
Amazing. So if these off-the-shelf T cells work, and you delete every CD7 bad guy and good guy in the body, you think there'll be any clinical consequences of getting rid of all the CD7 positive normal cells?
So this is the issue: if can people withstand long periods of profound immunosuppression, because it would target both the NK cells and the T cells, the recipients. So this would be one of those areas where a bridge to a transplant would seem quite rational to me, because I'm not sure if we can hopefully eliminate all these cells long term. And if they persist, which is one of the issues, then I think long-term survival without Ts or NK cells is going to be a problem. I think for sure you can live for a while. We have examples of that. And so patients can live many months without T cells and NK cells. It's probably years it's going to be a problem. So I think they could be a very good short-term therapy and a bridge to transplant.
Okay, so to finish up, I'm going to ask each of you, what do you think the most important clinical research trial or trials that are being done in your area. Dr. Daver, using checkpoint inhibitors in myeloid malignancies, what's the one or two things you think really need to be done soon?
Yeah, so what we're right now clinically most excited about is the combination, the immune checkpoints, Nivo-Ipi with AZA. So we have some early data showing very good activity, especially in people who have extramedullary disease, but also in bone marrow responses. But again, toxicity management is key. So I really think that that's something we're focusing on in the early salvage and then in the front line in AML and MDS.
You think AZA versus AZA plus AZA Nivo-Ipi?
Yep, I think that's going to be, the way to go. Now, there is also a question about can you combine it with venetoclax, because we know AZA venetoclax is good. We're seeing AZA Nivo high frontline response rates, and so that's another approach. But for now, I think we're going to focus on the double checkpoint, try to figure out management, toxicities, responses, and then see where to go next.
Dr. Frey, what's the next big thing in CAR T-cells against CD19 positive leukemias?
I think using it earlier in the course of the disease when you are at high risk for overall chemotherapy-dependent treatment failure because you're MRD, I think that would be great, and combining it with other immunotherapy, whether that's dual-targeted CAR T-cell therapy or a targeted antibody.
So the toxicity should be less if you use it early in the disease.
Yep, less toxicity and hopefully
Do you have any trials that are going to ask that question?
Yeah, so we have actually a trial open asking that question, small study though, so I think it would be great in that setting, especially for a dual-targeted.
Primary endpoint of that study is
event-free survival. Mm-hmm.
John, your work is preclinical so far, but when do you think it'll be in the clinic? What type of a trial will you do initially?
So I think the first thing is just to do a proof of principle phase 1 study in the clinic probably in the next 18 months to start. These are rare patients, as you know, so it will go probably relatively slowly. But I mean, the real issue is to see if it works and if it's tolerable. But I want to throw one other issue out that we're neglecting for immunotherapy, and that's the issue of AML. In addition to checkpoint inhibitors, when we think of bispecifics or CAR Ts to AML, this has been an incredibly difficult nut to crack. And I think that, again, somebody has to come up with a very innovative outside-of-the-box approach. One of Noelle's colleagues is doing this by actually trying to CRISPR out the myeloid target and then doing a transplant, and then, But I think that kind of really outside-of-the-box approach AML is very tough.
Right. CD123 has been one of the targets that was abstracted at ASH from City of Hope. Looking at that, there's CAR T-cells focusing on the same thing.
Yep.
So it's an exciting field in AML, with all the targeted agents we've talked about earlier today, and now with immunotherapy. Yeah. It's very exciting, a bit daunting, but I think we're going to benefit patients in the near future with these therapies. Thank you very much.